Another of the things you're going to do today is use a micropipetter, which is what this little guy is. A micropipetter is just something that dispenses small volumes of liquid. And you can control how much it dispenses by using these little knobs up here to turn, and you get a little numbers in this window and they tell you what volume you're actually using. This is where a pipette tip attaches. And then this is the actual plunger that lets it come up and down. So for example, when you add a tip and you wanted to take some liquid up and you have it set at the right volume, this little trigger has a couple of different settings. If you push down on it, it stops at a certain spot, and you push a little further and it goes a little bit more. Okay, and those are important for two different things. When you're going to take a liquid out, though, and going to move liquid around, you put your tip of the pipette on. Here's these little micropipette tips. You just take this and give it a shot like that, and now this tip is on there. You then would take this plunger and push it to the first stop. Take this tip, put it into your liquid sterilely, or whatever that's going to be. Gently release it up. Okay, notice I didn't do this. I gently released it. Then you pull it up and you'll have a volume of liquid in the tip. You then take this and put it into the next tube you're going to transfer to, and to get all of that liquid out, you push down to the first stop and then that little extra piece, and that shoots that little tiny bit out. This thing also has a little trigger, this little thing here, that actually if you push down on it, it'll shoot off the pipette tip. So let me show you how it's done on the samples that you're going to use. Okay? You're going to have two test tubes. You're going to label them with a Sharpie so that you know F1, F2, whatever you want, A and B, doesn't make any difference. But you're going to transfer some of this liquid from this one over to this one, and then back and forth. The idea is you want to try to keep these clean and use sterile technique. But to do this, you first will turn on your Bunsen burner. You will set this to a certain volume. I suppose we'll set these at about 500 microliters would be fine, or even 1 mil. For this demonstration, this one's only set to 100. You take your tip, put it in. This is all sterile now. Take your top off carefully, flame the glass, depress the tip or the plunger to the first stop, go down in, gently release, pull it back out, flame it, close it, take this one, remove the second cap, flame it, put this down in, push it down, a little extra, release, flame, cap it, and you're done. That's all there is to it. The idea is to keep these sterile, and so you and your lab partner can go back and forth and try to transfer liquid and not contaminate the solution.